Hello, and welcome to lecture number 17 in our cognitive neuroscience series, finishing up cognitive neuroscience methods, in particular finishing up functional neuroimaging. Be sure to watch part 1 prior to watching this particular video if you're not familiar with neuroimaging, because there's some important information in that first lecture, including an introduction to functional neuroimaging and experimental designs in this field, which is very important for understanding some of the terminology that I'm going to go through here today. So first off, we have positron emission tomography. Sorry, let me back up a sec. We're going to cover positron emission tomography, functional magnetic resonance imaging, magnetic resonance spectroscopy, which is something we haven't talked about at all. We've talked about these other two in previous lectures. I want to finish up with some discussion of limits of functional imaging. So let's start with PET scans. And essentially, PET scans, as it says, stands for positron emission tomography. So we are imaging these positrons that are emitted by radioligands. And essentially, what happens is a radioactive chemical is injected into a participant. It's a mild dose of radiation. It's about what you might get in a transatlantic flight. For those of you who don't know, when you fly at very high altitudes, as you do when you're flying, say, to Europe, there's a great deal of cosmic radiation involved in that, and that's about the dose you get when you are in a PET scanner. One of the things, and we'll talk about this in a moment, that is a problem with this particular technique is those radioligands generally have to be manufactured on the premises or very nearby because they rapidly decay, and so you couldn't order it from Amazon and have it arrive, a week later, because there would not be enough of the radioactive ligand left. So let's dive right in. Participants are injected with that radioligand, which binds to specific molecules. This can be glucose. This can be oxygen or oxygenated hemoglobin. Can also be attach itself to specific dopamine receptors, specific serotonin receptors. Also very useful in diagnosing cancer because there are proteins on the surfaces of cancerous tumors that those radioligands can attach to. So again, hemoglobin, glucose, neurotransmitter receptor sites are all targets for PET scans. So this is a broadly used technique in cognitive neuroscience because we can track where the brain is active at any given time, which is what makes it a functional neural image, because we can image where the brain is using any of these particular substances at a given time. But it's also uniquely suited for some studies focusing on specific neurotransmitters, and we'll also see magnetic spectroscopy is also useful in this realm as well. But in particular, PET scans are useful for binding to neurotransmitter receptor sites. So we can actually look within a living brain, or in vivo as we say, where in the brain those specific neurotransmitter systems are located, and also look and see whether or not across different participant populations there is different distributions of those neurotransmitter systems. So for example, we might see a difference between ADHD children and non-ADHD children. We might see differences between autistic children and non-autistic children. But we could also then look at the density of those receptor sites and how they're associated with specific functions in the brain. So if we're interested in something like working memory, we might then look at the density of the dopaminergic neurotransmitters in the prefrontal cortex and see if we can predict working memory based on the density of those neurotransmitters, something like that. One thing, we tend to limit each participant to 1 scan per year because of that radiation dose. One of the disadvantages of PET scans is that limitation. So we can't scan subjects repeatedly. We can only do 1 scan. Whereas with magnetic resonance imaging, you can scan all you want. So you can scan a person quite a few times. So give you a look at the kind of study that might be particularly suited for a PET scan. At the top, we have a magnetic resonance template where we're taking a brain, as we talked in the previous lecture, these idealized brains, which we can then place some of these maps, these specific types of parametric maps on that brain. And so in the middle line here, we have the average sort of healthy brain and density of cannabinoid receptors. And then we have the average PTSD sufferer, somebody suffering from PTSD, and their average brain, and the density of cannabinoid receptors. And what you can see is there is a significant increase in the density of cannabinoid receptors in the PTSD patients compared to the healthy patients. Now, of course, it's possible, there are two things that are possible here. One is that somehow trauma has led to an increase in the number of cannabinoid receptors, or, and I would say this is probably more likely, there's something about those individuals who suffer from PTSD that made them more susceptible to PTSD to begin with. So it might be the presence of those cannabinoid receptors that makes them more susceptible to PTSD than other individuals. So this is going to, this kind of research really helps us to try to investigate a variety of phenomena. So we can start looking at genetics and receptors and all sorts of combinatorial methods which allow us to investigate these kinds of phenomena and these specific hypotheses. So this is a nice scan, much more detailed than you often get with these kinds of images. But here you can see dopaminergic D2 and D3 receptors in the caudate. And really shows you nice lighting up, in particular, the density of these dopaminergic receptors in those basal ganglia, which are responsible for motor functions and also the reward pathway, nucleus accumbens. You can see the substantia nigra here at the top of this right, sort of the midline of the right bottom scan. Really, really fascinating kind of image. But these are the kinds of images you can get. So they give you a lot more information than, say, just an MRI might. So importantly, we can also use PET scans as a diagnostic tool. In fact, their primary use in this current era is in scanning individuals for cancer relapse. But importantly, it's a brand new tool in diagnosing Alzheimer's disease. And this is something that I'm personally interested in, as my mother has fairly advanced Alzheimer's disease. She's in a nursing home, doing quite well. I will want to add that. Memory is terrible, but she does quite well there. And has not had this particular scan done because there's really no reason to, and that's one of the things I want to get into. So Alzheimer's disease causes a buildup of both tau proteins and beta amyloid plaques. And we're at the point now that you can do both blood testing. You can certainly do an invasive spinal tap to test for proteins in the cerebrospinal fluid of individuals suffering from Alzheimer's disease. So those tau proteins can be measured. You can also measure the beta amyloid plaques via this particular type of PET scan. One of the important things to understand with Alzheimer's is typically we can't diagnose an Alzheimer's patient until it's post-mortem by looking for those beta amyloid plaques. So this PET scan allows us to do that. So one of the things with Alzheimer's disease is it's a part of a whole sort of spectrum of dementias. And so Alzheimer's disease and vascular dementia are the two primary types of dementias. And really at present, we can't differentiate one case from the other. There are some behavioral differences, and so we can make a pretty good diagnosis based on behavior, behavioral patterns. But other than that, we're kind of left with this seems like vascular dementia, that seems like Alzheimer's. It's very clear my mother has Alzheimer's. But the definitive diagnosis is the presence of these beta amyloid plaques. And so we can actually image these using what's called PIB, which stands for Pittsburgh B, I believe, is where the PIB comes from. I kept wanting to call it PIG when I was writing this lecture. But using this particular radioligand developed in Pittsburgh, that plaque can be imaged. Now, we don't generally do this in patients. This is actually only used in research settings. And the reason for that is there's no reason to. There's no treatment for Alzheimer's disease that's any better or different from how you would treat vascular dementia. And so there's no reason to go through an expensive scan when there's no benefit to the patient. And that's one of the things we have to always think about with this kind of diagnostic tool, particularly with Alzheimer's disease, is if there's no benefit to the patient, there's no reason to do the scan itself. If it's not tied to outcomes, then there's no reason to. A lot of us feel more comfortable having a definitive diagnosis, and maybe that can provide some comfort, but aside from that, there's really no reason to do this kind of scan. I also worry, particularly with the blood tests that are associated with tau proteins, that individuals who are relatives of patients with Alzheimer's disease will try to get these tests, and then that will cause stress and actually probably put them at greater risk for other conditions, including Alzheimer's, because it is associated with long-term stress. And so this is a great research tool. This is not something you want to drag your parents and grandparents down to get this kind of PET scan, because there's nothing we can do to treat them at this stage. We might get there eventually. And again, a good research tool. So for example, we're starting to try to figure out ways to clear that beta amyloid plaque. And so for example, if we could do that and see that in these scans, and we can see if that has any sort of benefit to the patient. So there are lots of ways in which this particular scan will be used in research, but not in clinical practice. And I think that's really important to note. Final note, the spatial resolution of a PET scan is limited. It's not as good as a magnetic resonance image or a functional magnetic resonance image, and it really differs depending on the radioligand used. And so there's no one-size-fits-all way to describe the spatial resolution associated with a PET scan. Also, they have pretty terrible temporal resolution because they have to, remember, we have to use blocked scans. And so we can't see any events unfolding over time. The best for that are the electroencephalographic tools we've talked about in previous lectures, event-related potentials, magnetic magnetoencephalography, etc. Final note, this is an expensive procedure. It's kind of hard to nail down exactly how much this costs, particularly for researchers, but my best estimate is about 3 to 5 times that of an MRI, because you have to have on-site a cyclotron and somebody to operate the cyclotron, so the staff and equipment costs are pretty dramatic. And also, of course, you can only scan one participant once a year, so you can't do sort of repeated measures designs in this kind of scan. So, we're talking about, I looked on sort of online, the retail cost of a brain PET scan is about $6,000. And if you're trying to run, and so of course it wouldn't cost a researcher that much. So let's say $2,000 a participant, just roughly estimating it. We have 20 participants, it's $40,000 just for the PET scans themselves, plus all the other research costs. So it's pretty expensive. Whereas an hour in an MRI unit, wholesale costs for a researcher is going to be a couple $100. So very different in terms of overall cost. So that gets us to functional magnetic resonance imaging, or fMRI. Don't ask me why the F is not capitalized. It's just how it is. So in functional magnetic resonance imaging, we're imaging what's called the hemodynamic response. And essentially, what we're doing is we're examining the ratio of oxygenated hemoglobin to deoxygenated hemoglobin. And we call this the blood oxygen level dependent effect. So the assumption is that where the brain has been metabolically active, where it's been doing stuff, as we say, it has used a lot of oxygen, and as a result, there should be more deoxygenated hemoglobin than in other parts of the brain. So that ratio of oxyhemoglobin to deoxyhemoglobin is this blood oxygen level dependent effect. And that's what we're measuring to infer that the brain has been accomplishing some sort of task in that particular area. So this bold effect is pretty important. Now, I do want to note that fMRI has much better spatial resolution than PET scans. And as we noted in a previous lecture, I believe in the structural imaging lecture, the spatial resolution of these scans, of fMRI scans, is going to be related to the strength of the magnetic field. So if we're talking about a 1.5 to 2 Tesla magnet compared to a 7 Tesla magnet, and they're developing an 11.75 Tesla magnet, so that spatial resolution is going to depend on the magnetic field strength. There is pretty rapid progress in this field, and there's actually a company that has developed a bedside MRI unit, so you can do actually a brain scan, a structural brain scan, right there at the bedside. It just plugs into a regular old outlet, which is pretty miraculous. So eventually I think we'll get to the point where we'll have this with fMRI as well. So here's the kind of images we might get. Remember, these are the statistical parametric maps in which we have taken these t-tests. So this is the APOE4, APOE3 region of interest. These are different genotypes in some particular task. I can't remember what this was. It doesn't matter. It's just illustrative. And so we have these statistical parametric maps for the task involved, and then in this last one, these are the SPMs for comparing these two particular genotypes. So this is a complex methodology where we're doing genotyping along with fMRI work, and then we've placed those differences in these voxels on this parametric map. And of course, you can see here we put it in a slice, versus here we put it on the surface of the cortex. And so you can see these areas where there's been differences in the functioning of these particular individuals. This is a study, for some reason, college students find interesting. The effects of cannabis on hippocampal activity. So when we get to memory, we're going to take a dive into this. So here we're just looking at sort of overall activity, and I can't remember what the task was in this one either. But we're looking at, and again, it doesn't really matter. We're just here to illustrate what these maps look like. We'll get into the details of these later on in the semester. But here we have controls versus cannabis users. And basically, it's not surprising. There is a reduction in activity. And you can see here in the prefrontal cortex, a little bit of hippocampal activity. Looks like motor cortex actually looks like there's some effects here. And again, what we're seeing are the effects of cannabis. So we're seeing reduced functioning in some of these areas and increased functioning in others, or increased use in others, not necessarily functioning. And we'll get into some of these issues. But again, this is what the goal here is, just to show you what these kind of scans look like. And what we're getting into. Then, of course, there would be more analysis of these particular regions of interest and a voxel by voxel analysis, etc. So one of the things I wanted to spend just a minute talking about is event-related fMRI, and I introduced this again in the previous lecture. And this is where we have actually time-locked scans to a particular event and lagged those scans in a way that we can actually see across time how individuals are responding to a particular task. So I wanted to find something interesting to talk about. So I found this pretty cool study on response inhibition and emotional impulsivity. And so for those of you in my Neuro class, we're going to be doing a go, no-go task as a homework assignment. Basically, in a go, no-go task, what happens is you push a button for some stimuli and you have to inhibit that for other stimuli. So let's say 99% of these trials would include the square. So whenever there's a square on the screen, you push a button. whenever there's a circle on the screen, you don't push a button. That would be the no-go tasks. So those are frequent versus the go tasks are frequent, the no-go tasks are rare. So basically, this is a measure of how good you are at inhibiting that response in the no-go trials. In this particular task, they've also added an additional bit where there are neutral pictures placed behind the go-no-go stimulus versus compared to an aversive stimulus. And there's a whole series of norms for these aversive pictures. So this is an example of an aversive one, because it seems to indicate a little bit of fear. And this is the reason they're doing this is they're interested in how this affects emotional impulsivity. So the task here is to inhibit a response, and that's what we're really interested in. So if we look here at the go versus no-go trials, and again, this is where we get that event-related type of fMRI signal, because we're actually able to, on a trial-by-trial basis, scan the brain of an individual. In here we are looking at neutral versus aversive. Let's see, response inhibition contrast. Sorry, this is in the comparing go versus no-go trials. And so what we get are, what's interesting about these is they're able to show increases in activity and decreases in activity. And so we see a reduction in activity in comparison in one area, and then you can see the change in that bold signal over time, that is the number of seconds. And what you can see is the go versus no-go trials. This is the left motor premotor cortex. No surprise there, because they're pushing the button here and they're not supposed to be pushing the button here. But then when we get over here into the posterior inferior parietal cortex in the anterior insula, we see significant increases in activity for the no-go trials. And what's happening here, this area of the brain is responsible for response inhibition. And you can actually see a little bit of it here, in which we are, this is a basic part of our attentional mechanism, our attentional networks, and we'll get into that later on in the semester. Anyway, so that's a quick look at that kind of go versus no-go trial. Then here we're looking at the emotional valence contrast. So we're looking at neutral versus highly emotional trials, and then you can see really getting some nice activation of the amygdala and some other areas as well. And then we can see this again on that trial-by-trial basis. Basic idea behind that is it allows us to look at how the brain's responding to some very specific types of cognitive phenomenon, and that's pretty exciting, at least it is for me. Now finally, and this is very important, there's been some question about the validity of some past fMRI research due to a glitch in the software, and there's some controversy over the controversy, as there always is. But I wanted to note that, and those of you who are interested in looking into it can read that Eklund et al. Article published by the Proceeding of the National Academy of Sciences. Very prestigious journal, which you should take a look at. So here's the source for that go-no-go trial. This is the source for the cluster failure, why fMRI inferences for spatial extent have inflated false positive rates, if you're interested in taking a look at that. So now, last thing to cover here is magnetic resonance spectroscopy. This uses nuclear magnetic resonance to scan for specific chemicals in the brain, so neurotransmitters, not the receptors like PET scans, but the actual neurotransmitters themselves. Unfortunately, because of the weak signals associated with the signals coming from these particular chemicals, we have to scan over a larger voxel, so that volumetric space. We use a larger size voxel, and it takes longer, so about 10 minutes to scan a single voxel. Now, by the time next year comes around, this will be, I'm sure, very different. Computer processing capacity is changing so rapidly, and technology changing so rapidly that we have to, think about how long this might take. But it's really interesting. We can actually see where the brain's using a specific neurotransmitter, and that's very exciting. gamma aminobutyric acid, is a frequent target for these scans. One of the most common neurotransmitters in the brain, but it also has a lot of important behavioral functions, and it's one we'll talk about a number of times throughout the semester. So it allows for some really interesting studies. This is one I found that's really fascinating. It's from Nature. And essentially, what this is about is how does the brain accomplish inhibiting unwanted thoughts? And inhibiting unwanted thoughts or controlling unwanted thoughts is a really important part of treating things like obsessive compulsive disorder or just overall treatment of using cognitive behavioral therapy is controlling unwanted thoughts. And so understanding the neurobiology of that is a really interesting and fascinating application of how cognitive neuroscience can contribute to treating individuals with mental disorders or even just basic difficulties. So here we look at GABA and its involvement in controlling unwanted thoughts. And so essentially what they're using here is a think-no-think paradigm, which is very similar to the go-no-go paradigm. So we're trying to inhibit unwanted thoughts. This comes again from Nature Communications. So I want to take a closer look at this. Is the overall figure that looks at a bunch of other issues that are beyond what we're interested in. And so we can look here. These are the areas that they're studying. And so that's one of the things I wanted to point out is we're not scanning the whole brain. We're scanning some limited areas around the hippocampus, prefrontal cortex, and the visual cortex to try to look at how GABA might differ across these experimental conditions, particularly in the hippocampus. And what they did find was that essentially GABA is what is inhibiting that thought, so that inhibitory neurotransmitter plays an important part in that. And that might provide us with some potential avenues for exploration for medications for treating anxiety disorders. We already know that anti-anxiety drugs such as Xanax, Valium, Ativan involve GABA. That is, they increase the function of GABA, what we call GABA agonists. And so that might be why they're useful in limiting those kind of intrusive thoughts. So pretty fascinating use of MR technology. So I want to finish with some limits of functional imaging. First thing we have to remember is this is all inferential. We're not measuring neural signaling directly. We're correlating and inferring that there is a change in the signals associated with phenomenon. So we want to use converging evidence from a variety of methods: neuropsychology, looking at individuals with brain injury, perturbations, such as using drugs or RTMS, something like that, to perturb brain function. Also, ERP evidence. All of these things need to combine together to provide us with that kind of information. And importantly, we're a long way from using individual scans. No two brains are really alike, and most studies focus on similarities, not exceptions, which might be a critical distinction. So looking at individual differences is where this research is heading, but we're not really quite there yet. So we have to really understand that whenever we're looking at these scans, we're almost always looking at scans involving groups of individuals. So I wanted to sort of finish with that thought. Now, trying to localize brain functioning is something we've been trying to do for a long time. It's a whole industry in the late 19th, early 20th century called phrenology, in which they thought they had managed to localize brain functioning based on bumps on your head. And of course, that turned out to be wrong. We could be just as wrong. So a little humility, I think, is important. I think these are great techniques. I think they're important. But I think we also are at an early state of understanding how the brain works. All right, thanks. We'll see you next time.